On behalf of KoreaBIO, Sidley Austin, and BioCentury, I wanted to welcome everyone to day 2 of our virtual global IR pitching session. My name is Josh Berlin, and I lead BioCentury business development team. If you don't know BioCentury, we help biopharma executives and investors make business-critical decisions through analysis, data, and business intelligence. We also help connect global industry leaders through our strategic conferences and digital events. We hope this inaugural event will become an annual showcase around JPM Week, helping to connect Korea and Western biopharma executives and investors. Hopefully next year, KoreaBIO, Sidley, and BioCentury can host you all in person in San Francisco alongside JPM. In the meantime, this virtual event is an opportunity to hear 16 selected biotech companies tell their stories to US, Asia, and global investors. Yesterday, you heard from our first 8 companies, all based in Korea. You also heard a great panel on cross-border opportunities from the perspective of US-based executives. If you missed any of the day 1 program, you can watch a recording on the KoreaBIO YouTube channel. Today, you will hear from 8 more biotechs from US, Europe, and Asia. I wanted to thank my colleague Hangjang Lee for leading the presenting company selection committee for today's program. Before we get to this pitching session, we will start today with our second virtual panel to discuss cross-border collaboration, this time from the perspective of Korea industry leaders. As you all know, biotech is a global industry, and cross-border collaboration is often a key to accelerating drug development to patients everywhere. I'm really looking forward to this panel in particular, as Korea has been a major focus for BioCentury over the last several years. Each November, for example, we host a Korea Biotech Roadshow track at BioCentury's China Healthcare Summit in collaboration with KIDI, the Korea Health Industry Development Institute. BioCentury also has a great partnership with News One to release translated versions of BioCentury's business intelligence to the Korean biotech community. So for BioCentury, we see this week's event as a continuation of our efforts to help connect Korea industry leaders to the global biotech ecosystem, and we are very honored to partner with KoreaBIO and Sidley Austin to help do so. So with that brief introduction, let's get started with today's panel. It is my pleasure to introduce our session chair and moderator, James Lee. As many of you know, James is a serial biotech entrepreneur and CEO of the Korea Biotech Bridge Biotherapeutics. I usually meet James in San Francisco during JPM Week, so although we cannot meet in person this time, it is an honor to share today's virtual stage with him. I will now hand the session over to James and we will get started. James, it's all yours.
Thank you, Joshua, for your very kind introduction. And I'd like to say Happy New Year to all of you on the video conferencing, and also welcome back to Global IR pitch, co-hosted by KoreaBIO, BioCentury, and Sidley Austin. Today, I have 4 panels who can give you information, insights, and recommendation to those who are considering cross-border collaboration with the Korean biotech, venture capital, government, or clinical development system. Let me introduce 4 of our panels from Deborah Chi, Ji-Ung Chun, and Sang-Hoon Lee, and lastly, Bum-Jun Park. And from Deborah Chi, would you introduce yourself briefly before jumping into some discussion?
Okay. Good morning, everyone. Good evening, everyone. I'm very glad to introduce the Korean clinical trial environment and the status today. I'm Deborah Ji. I'm working for BridgeBio as external director at the moment. And also I'm a freelance consultant for Korean companies and foreign companies to help them to conduct their best clinical trial program in Korea and in Asia. I worked for global companies for 20 years, and from 2014, I had been in charge of Korea National Enterprise for Clinical Trials to build clinical trial capacity in Korea. Thank you.
Okay, and representing Korean venture capital community, Jiung, would you introduce yourself?
Yes, thank you. Hello, my name is Jung Chung from KTB Network. KTB is the first cross-border VC, Korean VC, since 1981. So our headquarters in Korea and has offices in China and USA Bay Area. Our AUM is over $1 billion US, and myself, I'm charged of life science investment in KTB, covering Korea and Greater China and USA. My major is pharmacy, and I'm the registered pharmacist. Prior to KTB as of a VC, I worked at Daiichi Sankyo as of a global clinical study manager and CKD Pharma as a scientist. Thank you.
And thirdly, representing maturing Korean entrepreneur activity, Sang-hoon, would you introduce yourself?
Hi, yeah, my name is Sang-hoon Lee, founder and CEO of KoreaBIO, located in Pangyo, South Korea. I have a US industry background, such as Chiron, merged by Novartis, and Genentech, Exelixis, and AstraZeneca. Then I came back to Korea and co-founder of the PharmEpsin and work for Hanwha. Now, I studied KoreaBIO 5 years ago, and we are developing a lot of bispecific antibody by collaboration in China, South Korea, and US-based company.
Okay, and lastly, representing Korean government's initiative, would you, Bum-jun, would you introduce yourself?
Good afternoon, and maybe some people, good evening. My name is Bum-jun Park. I work at KOTRA, which is a Korea Trade Investment Promotion Agency. It's a government agency for trade and investment promotion. And my role at KOTRA is investment project manager for life science and healthcare industry. I recently finished the 4-year term in our Singapore office. And also KOTRA has a global network, and which includes 9 offices in North America. And now, today, discussion session, I would like to share Korean government initiative as well as government incentives.
Yeah, great. With those introduction, let's go to topic. Firstly, about the clinical development environment. Deborah, you have been working in the Korean clinical development ecosystem for more than 2 decades at the private sector and also public sectors as well. So what's your observation, and you have been seeing in Korean clinical development ecosystem from hospital system perspective and regulatory perspective and the researcher's perspective as well?
Okay. I will start from the regulatory environment. The Korean GCP was legislated as early as in 1995, and the ICH GCP was adopted by Korean FDA. Now the name is MFDS back in 2000. The IND process was introduced firstly in 2002 in Korea, and the Korean government has continuously invested for past 15 years in clinical trial capacity building. All of this led the unprecedented growth in quantity and the quality of Korea clinical trial over the past 2 decades. With all these efforts, Korea has been one of the top 10 countries in terms of the number of clinical trials conducted in Korea in the world. And Seoul has been the #1 top city in the world in terms of clinical trial density and even the absolute number of clinical trials. And multinational phase 3 studies sponsored by global pharmas and Phase 1 studies by domestic companies have driven the growth, and phase 1 studies by Korean companies are growing everywhere. That reflects the activity of Korean companies in early phase clinical development. For example, 168 phase 1 studies were initiated by Korean companies in 2019 only. Those studies were conducted in well-equipped phase 1 units built in major university hospitals by the extensive investment by Korean government. And the foreign sponsors like the speed and the quality they get from performing trials in South Korea as well. The population density of Korea ensures a strong availability of patients in most major indications. And the Korean healthcare system provides universal coverage through technologically advanced hospitals concentrated in large cities. Rapid recruitment and the study startup are enabled by optimized recruitment practices by hospitals and the large volume of daily patient traffic. The investigators are very experienced in major therapeutic indications. Top Korean investigators in cancer studies have taken the lead investigator role and also advisor role for global pharmas for their development plan, not only for the Asia prevalent indications such as gastric cancer and the liver cancers, but also major indications such as lung cancers. Above all, Korea has streamlined the regulatory process for clinical trials. The IND process was put in place to foster faster study startup times with a goal to get the trial approval within 30 calendar days from the submission at the MFDS. Especially the procedure allows sponsors to simultaneously submit trial requests to ILBs, the institutional review boards, and the MFDS, thereby reducing the time needed to get the approval. So for your clinical trial for foreign market, the availability of a competent CRO is very important as well. Around 40 CROs are operating in Korea, and most major global CROs have their offices in Korea as well. So among the top 10 CROs operating in Korea, 6 are Korean CROs, and they have a comparable capacity and experience to global CROs. That's it.
Okay, thank you, Deborah. You have covered regulatory system and hospital system and quality of investigators, and also very important component of CROs. So you see, do you expect incoming first-in-human study will increase as time goes on due to such an excellent Korean clinical development ecosystem?
Yes, actually, the number of phase 1 trials by foreign sponsors has been increasing. In 2019, we had around 50 phase 1 trials by foreign sponsors. Most of them were the patient-oriented studies, such as cancer phase 1 studies. But still, the PK components are included, and the excellence of Korean phase 1 units contributed for those studies as well.
Okay, thank you. So, so with that, if some non-Korean companies are considering first-in-human study in Korea, so what's your recommendation in terms of the contact and approach?
You can contact the clinical trial centers that is the coordinating office in each hospital. And also they have their own website for their phase 1 study unit. So you can easily find the English website, for example, the Seoul National, Yonsei Severance hospitals. Also, you can contact me. I know all of them very well, and I can give you the guidance. So you can reach me through James.
Okay, thank you. Anybody who are interested in the Korean first in human study, please reach out to Deborah or myself, or the organization called KoNECT. Okay, let's move to next topic about the venture capital activity in Korea. Ji-Hung, you have been in VC industry for more than 10 years and should have observed some changes in Korean venture capital community. Originally, it has been known as a truly domestic industry, but I see now more and more Korean VCs are coming out to find a investment opportunity not only in Korea but also in US, Europe, and Australia as well. So please give your thoughts and insights to those who are interested in interacting with the Korean venture community.
Yeah, thank you, James. And so it's the first of all, I want to say it's all about our government or some entrepreneurs such as James or the Sang Lee. Those guys are actually some trying to improve the rate of a Korea biotech's reputation in the world. So that's why it helps us a lot for us as well as of investors to jumping in the cross-border deal. Yeah, so let me introduce briefly about some biotech ecosystem, investment ecosystem in Korea, and then want to tell about some cross-border activity from investor side. Especially last year, 2020 was a year unlike any other. So COVID-19 has led to the fruition of trends that have been long waiting, such as mRNA vaccine or diagnostics for COVID-19, especially in Korea. So although there is a slight reduction of investment, biotech investment in Korea at the first quarter of 2012, but the investment atmosphere is back later. So according to BioSpectator, approximately over 130 bio companies have received over 3.2 billion the financing, including IPO and listed company. And at the venture stage, there are 300 million for Series A, 380 million for Series B, around 300 million for the Series C and pre-IPO. But this data is based on the public announcement or news release. So I do believe that the investment size is much more larger than that. But but a few years ago, recently biotech investors in Korea is getting larger and harder and improving their own expertise. When I joined KTB about 7 years ago, there is only 30 to 40 life science investors in Korea. But nowadays, at least over 100 biotech professional investors are working in Korea now. And also in regards with cross-border investment from Korea, around 2014 or 2015. So actually, I did investment alone in case of KTB's cross-border investment. So such as for the USA or Greater China. But currently, there are at least over 10 cross-border VCs, 10 cross-border Korean VCs to investment in biotech in abroad. For example, in case of back to the 2014, it's really hard to make the syndicate cross-border investors in Korea. And in my case, I invested alone at the biotech in Boston in 2014. But last year, the 6 financial investors, including KTB and 1 strategic investors, have invested in biotechs in Bay Area, Genetic. Maybe yesterday they are the CEO is presented. So not only venture capital, cross-border venture capital in Korea, but the many Korean pharmaceutical company have started to invest in biotech in abroad for some open innovation. And maybe last but not least, let me introduce about our Kosdaq market briefly. And then Kosdaq exchange has a very good and development IPO system for free revenue biotechs. And over 80 biotechs have been listed in by this pre-revenue track since 2005. This track for the biotech has been very successful to raise enough funding for biotech before and after IPO, as long as their fundamental and the catalysts are good, and has made our investment community and biotech ecosystem much more fruitful. So to compare with Hong Kong and Shanghai star market, so we have a much longer history and then larger experiences. Yeah, thank you.
Okay, thank you, Jiyoung. So you have a really nice overview about Korean change in Korean venture capital community and also a Kosdaq market. Too many Westerners, Korean market, especially Kosdaq market, has been known to give a really high, sometimes crazy valuation to biotech companies, and also at the same time, it is known to be very from the investor side. Most of the investors on Kosdaq market is retail investors, uneducated and very speculative. Do you see some changes in investors' behavior in Kosdaq market, especially for biotech companies?
Yes, I do believe so. Since we have some longer history than the except the Nasdaq for the biotech investors, so maybe you guys maybe some of you may remember that the 5 or 6 years ago, there is if one pharma or biotech company has a good news, that it impacts all of the other biotechs or listed companies, the valuation, whereas the price at the market. But however, so the investors, even the retail investors, they recognize that. So what is the real value, and then how catalysts can affect the market? So recently, I do believe that it's not affected by only one news, or they are more focusing on some fundamental the biotech has. That is big change, and also another big change is that not the professional investors such as some pharmaceutical company or some general companies. They start to understand about the value of about the investment. So those guys are coming in, and then with a big ticket, and actually it also helps to stabilize the market itself. That's a huge change.
Right. So your observation is really in line with my observations as well. I see some very professional institutions and some high net worth individuals, and also other type of investors are now looking the biotech sector as a really long-term and also very maturing industry sectors. So we will see more educated and long-term viewed investor will see biotech sectors and also very promising biotech companies as a really nice investment opportunities. And let's go to innovation and entrepreneurship and cross-border collaboration from biotech perspective. Sang-Hoon, you have been working in the industry for more than 2 decades at many US companies and Korean companies as well. So what's your thoughts and recommendations to our potential cross-border collaboration biotechs into Korea? What's the pros and cons of working with the Korean biotech companies?
Yeah. Actually, as you know, Korea has been known as, the growing, the maturing country for putting a lot of effort for innovation. Like China, both, the Korea and China had tremendous, the biotech, the form. Especially China is more, the Chinese came from US-based, the biotech and pharmaceutical company. Actually, the Korea also has couple, the many, the company form, and innovation is one of the way. In my mind, I think the to move forward to US, the industry biotech. A lot of Korean company has been working with the Chinese company as a cross border, and couple, the good example. Recently, the one of the good ADC technology company, LegoChem in South Korea, decided to have a collaboration with a Chinese company called KoreaBIO to develop ADC, the long-term collaboration. And also, I mean, the Green Cross, the set up some, the Cambridge to license out the entourage for commercialization and clinical development in China. So I think that it'll be interesting to see additional cases. I also searched, the Celtrion recently set up the joint venture called VCell Healthcare for commercialization of the biosimilar. And also, the Samsung BioApp is also, the working with the Sidley, the healthcare, and to commercialize the biosimilar. So I think not only biosimilar, but also innovation. We have a lot of, the cross-border cases. And I think the within, in also, the between Korean company, we have a lot of open innovation. And one of the cases is the LegoChem. My company, KoreaBIO, worked together last, the 3 years to develop one of the ADC project, and we recently licensed out to Keystone. So that's, the very interesting cross-border. I hope to see more, the cross-border collaboration between Korea and US-based company, Korean and other Asia company other than China. One also, I've been, the working with KoreaBIO has been working with the IMAP, NASDAQ, the company. We decided to collaborate for developing minimum 304, the bispecific antibody. Although, during drug discovery program, 2 projects actually terminate. However, 2 projects move forward very well. So one project we identified last December, and then we're gonna do another one on February for US IND. So I think the eventual goal is both Chinese company and Korean company work together, and then, do the US IND, since, we, KoreaBIO has no, the entire, the capability to run clinical trial by ourselves. So we actually find the cross-border collaboration. And I think, not only collaboration and co-development, KoreaBIO also invested on IMAP as a strategic partner when they do the IPO. So actually stock price went up like 4-fold compared to when we invested. So I think a lot of, the opportunity between Korea and China, between Korean and many other companies outside Korea can be worked together. I hope to see cross-border collaboration expand in near future. Thank you.
Okay, great. Very concise and nice coverage. So traditionally, most of the cross-border transaction between Korea and Western was a very straight licensing. But recently, we see more collaborative research or joint development or investment are getting popular and popular. So we see more creative collaboration between Korea and Western as a cross-border collaborations. And lastly, we have Bum-Jun to hear about the Korean government's initiative to facilitate the cross-border collaboration between Korea and Western or Chinese companies. You have roughly 4 minutes, but I believe you will give a really concise and thoughtful explanation.
Okay, thank you, James. I'd like to start with sharing the Korean government initiative, which is published in 2019. So the South Korean government also understood the importance of the biomedical and also healthcare industry, and also government announced a big roadmap to promote and boost the industry, which includes utilize the big data, expand investment in policy financing and R&D, and also improve the regulation to match the global standards under a strategy to create an innovative ecosystem ranging from technology development, approval, production, market launch of the biohealth industry, which is biopharma based. One of the important part of the government initiative is the increase of the investment in R&D sector. In 2019, South Korea government invest about 2.5 billion US dollar, which will be this will be increased to 4 billion US dollar in 2025. As the government investment and also the collaboration, the effort increase, I guess there will be more opportunity to collaborate with other countries. And thanks to South Korean government initiative and also the South Korean companies' enthusiastic effort, the biohealth industry set a new milestone when it comes to the export. The biohealth industry is counted as a top 10 export in South Korea. And last year, the pharmaceutical export totaled 14 billion US dollar. As the sector of the Korean export fell about 5.4%, but this pharmaceutical export, the volume is up about 54%, which is driven by the biosimilar products. Some of you already mentioned about the biosimilar industry. South Korean biosimilar industry is one of the key industries in South Korea now. And there are two leading players, as you might heard of, Celltrion and Samsung Biologics. There is one interesting part that those two companies are also started as a foreign investment company. Celltrion attract investment from the Singapore's sovereign investment Temasek Holdings, and also Samsung Biologics. They start with the joint venture with the US company. There are also the cash grant, the government incentive to promote and boost the foreign direct investment. These, especially the biomedical and the healthcare industry, which will be the new growth engine for South Korea, is one of the key industry can get cash grant from government. Yes, that's from my.
Yeah, thanks, Bong-joon, for giving us a very informative hint about how Korean governments are really eager to boost this sector. So so we are very close to the end. So all the panels are really informative and also time conscious. Backed by growing innovations and increasing and growing venture capital activities and government support, and also maturing clinical development environment, I believe cross-border collaboration between Korean ecosystem and Western and Chinese ecosystem will surely move to next level in coming several years. So since we don't have any further time, any question or so Carrie can go through our organizers, KoreaBIO, Sidley, Austin, or BioCentury, or you can reach us directly through email. So in summary, so I believe Korean and non-Korean collaboration will increase very rapidly, and I hope to see some more non-Korean companies can present at next year's conferences. Thanks for panel and audiences and organizers. Thank you.
James, thank you very much for that very interesting panel. This is Josh Hofheimer speaking from Sidley, Austin. That was a terrific panel, and thank you to all of you all who were presenting there. I wish that we could be there in person with you, but hopefully we'll be there because we've been with KoreaBIO for a number of years in person, and I've enjoyed spending time with you all and learning more and more about the different Korean life sciences companies and opportunities to work together cross-border. But hopefully that will be remedied soon. And we'll be able, this time next year, we'll be able to all be together either at JP Morgan in the US or at a KoreaBIO event in Korea itself. So I'm going to get us moving into the next panel without much ado. My name is Joshua Hofheimer, and I'm a partner at Sidley Austin, which is a global law firm, almost 2,000 lawyers in 20 offices around the globe, and specifically 6 offices in Asia. We are very committed to global life sciences, and I have been working for a number of years on an initiative that focuses on representation of Korean life sciences companies, doing transactions, deals, and collaborations with both global biopharma and regional cross-licensing deals with China especially, but also with companies that are partnering with or investing in Korean life sciences companies. And that's part of the global platform that Sidley offers. My colleagues and I are very committed to this sector. We think that the Korean life sciences market is fantastic, and there is some great innovation coming out of it. And I'm especially pleased to be able to introduce everybody on this pitching session today, and the fact that we have a number of foreign companies sharing their expertise, their experience, their products, their therapies with Korean partners, Korean investors, and the like. I think is very exciting, and it's sort of part of the beginning of the next stage of development and maturation of the innovative Korean life sciences market, in that you're attracting small or smaller proprietary and innovative companies to events that you're hosting, to activities and to opportunities to create exposure and connection with the Korean market, and not just going beyond sort of the global bio partners looking to either acquire companies, perhaps set up manufacturing and supply arrangements, but really leveraging the interesting innovation that's coming out. So without further ado, I think our first panel, and I see Susan Dillon, you're there ready to go. She's the CEO of KoreaBIO Therapeutics. I don't want to hold this up any longer, so Susan, please take it away.
Thank you very much. And thank you for the previous session. I enjoyed it very much. Can you see my screen? Yes. Okay,
yes.
Great. Okay. So again, hello, everyone. I'm going to tell you today about our company, KoreaBIO Therapeutics. We are focused on creating a new class of receptor-targeted genetic medicines. And just to introduce Arrow. So here are several highlights describing Arrow. We are focused on a novel protein drug platform called Centurins that we believe can unlock the full potential of various kinds of RNA-based medicines. We have a robust drug discovery engine, which consists of Centurin RNA drug conjugates. We have highly differentiated features of Centurins and many advantages versus monoclonal antibodies and other non-specific targeting mechanisms. Our early pipeline at Arrow is focused in orphan genetic diseases and orphan immune diseases. And we have established a partnership with Ionis Pharmaceuticals about 1 year ago Ionis are the world leaders in RNA medicines focusing on antisense. We have an extensive patent portfolio and a very strong IP position, with many issued patents and others that are in the provisional stage. We have built a very experienced leadership team in the 3 years since we founded Arrow. My background is in drug discovery and development. My most recent role before I co-founded Arrow was as the global therapeutic area leader at Janssen for immunology. I worked for many years developing a variety of monoclonal antibodies shown for autoimmune disease. My co-founder, Ken Neal, a well-known antibody engineer, and she has applied her protein engineering skills to the discovery of centurins. She is co-inventor on all of the centurin patents. Scott Kinberg is our Chief Business Officer, has extensive experience in prior companies such as Celgene and Roivant. The rest of our team has expertise in antibody drug discovery and translational research, and in chemistry and manufacturing of protein drugs. So we are very well positioned for both the discovery and the development of this new class of biologic medicines. We very recently completed a Series A financing. We announced this in January with several high-quality investors and a full round of $88 million, which provides us the runway over the next several years to take our early pipeline and advance it into clinical development. Let me talk about centurins. So centurins are a proprietary and binding platform. Centurin small proteins, as shown in this picture, of about 10,000 milliliters weight, much larger, 1/15 the size of a typical monoclonal antibody. Centurins have exceptional stability and solubility. We can express them in E. coli, and this is our manufacturing strategy for clinical development, and have identified ways to specifically attach centurins to specifically attach RNA drugs and other drugs to specific sites within the centurin. We are focused on finding ideal drugs for bringing the RNA medicine into the right tissue. And our work to date has focused on centurin antisense conjugates, abbreviated ASOs, centurin silencing RNA conjugates, siRNA. These are different RNA drugs. One is single-stranded, the antisense siRNA double-stranded, but the synergate works in a similar fashion. So we identify centurins that are specific for a cell surface receptor on target cell. We chemically covalently conjugate siRNA or the antisense, and on binding, this gets internalized, and ultimately the RNA drug can get to the site of action and inhibit the production of messenger RNA that encodes a specific protein. So it is the inhibition of disease proteins, of disease-causing proteins, that underpins the mechanism of action of our drugs, with the very important distinction being that we are able to target these drugs to the right tissue. We have data, and we firmly believe that our platform of centurin oligonucleotide conjugates are going to optimize efficiency and delivery and have many advantages over antibody complexes. In short, we have a very rapid discovery process, flexible platform, and a very simple methodology for conjugating centurins and RNA, as well as a scale-up manufacturing approach. At the end of the day, we believe that our drug conjugates will be more effective, safer, and less costly to produce with excellent IP protection. We have been focusing on identifying centurins to a particular receptor called C71, which is a transferrin type 1 receptor, which is present on muscle cells and heart, as well as other cells in the body. We've developed compelling in vivo knockdown data, meaning gene mutation data, with centurin RNA conjugates, centurin antisense conjugates, much improved over naked unconjugated versions of these RNA drugs. We've shown that this effect is quite durable and has a very high tissue selectivity for muscle versus either liver or kidney, which are the typical tissues where siRNA or antisense accumulate. Given the exciting data that we have generated in animals, we're focusing our early pipeline on centurins that specifically bind to CD71 related to these different types of oligonucleotide drugs. This is a snapshot of our pipeline. We have 2 current programs at Arrow that are in the lead optimization phase, one for genetic muscle disease, is for orphan autoimmune. In our collaboration, BioCentury, we have 3 current programs. The first one is in a different genetic muscle disease, and 2 other programs. All of these are centurins conjugate antisense oligonucleotides that have been discovered at BioCentury. This collaboration that we established about 1 year ago with BioCentury is aimed at solving this challenge of extra targeting of antisense oligonucleotides. This as a very critical transaction for Arrow, as we are a young company, and by virtue of this relationship, we have a way to de-risk our platform by working with a world-class leader in the field, of course, strengthen our financial position, and afford us the opportunity for future pipeline assets. At the same time, the scope of the collaboration is quite narrow. BioCentury only have a certain number of targets that they can work on. The progress has been really outstanding over the past year. We identified conjugates that give very nice in vivo activity, providing the first proof of concept. BioCentury have exercised the option against the first program. We announced this late in October, and we are working towards the preclinical development candidate in 2021. There are many different applications of centurins. I've talked about the application about conjugate medicines. However, we have also developed data for conjugation of centurins to nanoparticles that contain all sorts of different payloads, as well as the potential for blood-brain barrier penetration, use in cell therapy, use as toxins, as well as use as bispecific or multi-specific drugs. I just want to make a mention that we have done a significant amount of work in oncology, although this is not currently our focus at Arrow. We are very interested in potential partnerships in this area. We have identified many different high affinity centurins against validated tumor targets. Some of them are shared. We have many against solid tumors, such as EGFR, PSMA, and many against liquid tumors, including BCMA, CD19, etc. I've demonstrated in vivo proof of concept in different disease models with these centurins, expressed them or formatted them as bispecific molecules that can target 2 different receptors on the surface of tumors, looked at toxin conjugates, siRNA conjugates, and have quite a broad IP portfolio in the field of. So I'll just end by repeating what I stated at the. Our focus is to leverage our novel centurin protein drug platform to unlock what we believe is an enormous potential in the field of RNA medicines, but where these medicines really face a challenge in terms of effective delivery to the right tissues. Have a robust discovery and many points of differentiation, monoclonal antibodies and larger proteins, an exciting pipeline in orphan, genetic, and immune diseases, a key partnership with BioCentury, leader in the industry, and an extensive patent portfolio. So thank you very much for the opportunity to present today, and I'm happy, if there is time, to answer any questions.
Can you hear me?
Yes, I can.
So just one quick question before we move on to this panel. Do you all have a strategy, a particular strategy that may vary disease with regard to the timing of when you anticipate partnering, or are there any therapeutics that you think would take all the way through like registration or beyond?
I had a difficult time hearing you, but I think your question was, do we have a strategy with regard to the stage at which we partner an asset or partner the technology? Is that correct? Yes. Okay. So I think that for at this point, we're a young company and we are very flexible. Partnering with Ionis is discovery stage. How we go for, I think that we're interested in moving our own pipeline forward. This time we expect that we will take all the way through, but there may be opportunity to get more to partner on commercialization. I answered your question.
Terrific.
I guess one more comment is that, again, with regard to oncology, we have quite a number of assets as well as a platform application to oncology. We have we decided not to focus on in this disease, and so because of that, we were very interested in partnering around our Centurion platform, and we would welcome any questions along those.
Thank you. Thank you very much, Susan. Hopefully you all can hear me. So, but just in case, I'm not going to play much. I'm going to hand over to Piers Ingram, who's the co-founder and CEO of Humming Biosciences, who's going to be presenting next. Piers, hope you can hear me, and take it away, please.
Hi, Joe. This is Piers. Speaking. Can you hear me? Hello, everybody. I think I ensure that people are able to see before I go ahead. And the sound quality is a little bit musty. Would that be kind of to whether you could hear me or not?
Yes, you can be heard.
Okay, that's great. Thank you, Josh. Okay, so hello, everyone. I'm co-founder and CEO of BioCentury. To organize this opportunity to present today. Very much enjoy the discussions. BioCentury is a enabled a company. A company 5 years ago, myself and co-founders really been applying the tools and systems of computational biology to drug discovery, and focusing on which we felt had really compelling biological stories, and yet being drugged with classical approaches. This strategy really focused on one topic which I'll speak more about in a couple of minutes, HER3. Pursuing that target also developed of a technology for antibody discovery, which now underpins a lot of our work as well. So I'll talk a little bit about that too. The company now has just over 50 employees, split to Singapore and the US. And we have last year raised our Series B financing, support of major Korean investors. So it's very much appreciated the support of the company, and it's a very exciting time for us as we take the first 2 programs into the clinic in the next years. So with that, I'll dive into the content. So the platform that we was something quite different to antibody discovery technologies. For those who are familiar with how classical antibody works, it's still very much a screening-based process. Typically, we try antibodies and characterize what they do first, and then when they bind. For many targets, also several targets where, because that binding approach didn't necessarily coincide with the functional regions, we not really have good antibodies that we could develop as therapeutic candidates. And so what we thought would be a more effective strategy was if we could find a way to apply some of the methodologies from rational antibody discovery from small molecules and try to put these ideas to the biologic space. And what we tried to do, I think, captured a little bit by this screen on the left, where classical antibody discovery approach, they give you many good antibodies to not necessarily functional region, very few antibodies to the functional region. We want to flip that around. So our technology platform consists of 2 core pillars. The first is the computational construct aspect, where we try to identify where that functional region. We don't necessarily know every where we want these antibodies to bind, although we may have a hypothesis. We really want to define that thoroughly. I'm seeing the message from multiple groups that the quality is really improved. The organizers can suggest how they'd like us to do. I think this quality has been quite challenging.
It appears there's something happening with Zoom, Piers, that I don't know if you can hear me. It's making it difficult to hear everyone.
Yeah, I prefer I can hear you as well.
Just pause for a minute and let's see if we can get a message. KoreaBIO has managed to remedy this. Here, we try now. Can you all hear me now?
It's still very broken now.
We're getting one of the organizers. Here's an issue with Zoom. So let's just hold off for 1 minute. The good thing is that you can hear me. We had an unfortunate incident, completely unrelated, a personal matter that happened to Prestige Pharma, BioPharma. So they're not going to be able to present, which will enable us to, if we need extra minutes here, and run up, we won't lose time overall program. There's a suggestion that maybe with so many attendees, but there's a suggestion attendees switch to YouTube Live, and then if analysts stay on Zoom, it may help them to free up bandwidth. So perhaps some of those attendees who have ability, if you jump over to YouTube Live, we'll see if that improves things. How am I coming through now, Piers?
I think it seems a little better now. Am I still clear?
No, you're still a bit choppy. If attendees could actually switch over to, go ahead.
No, I was going to say I stopped to see if that improved the bandwidth issues at all.
It does look better when you stop video. Can you try speaking now?
Sure. Let me know if this is better and is viable.
Yeah, I think that's better. And there was a huge streaming link that was recirculated in the chat, so people can on that and rejoin there. Piers, why don't you start from the beginning and just sort of quickly go through the introduction and then continue forward. We're actually, because of the loss of Prestige, we won't be behind in the overall timing.
Sure, I have to do so. So, starting quickly from a recap of the introduction, Hummingbird is a company founded by myself and colleagues in 2015 with an idea of applying tools of computational systems biology to drug discovery. We've been extremely focused on targets with we believe has a compelling biological story and a degree of validation, but which has not really been effectively drugged by other platform technologies. And we feel that our approaches can deliver an opportunity to progress. We initially focused our efforts on, and I'll speak more about that program in a moment, but the development of that program, we also developed a technology platform in order to overcome the challenges of drug discovery, which has become an extremely powerful technology for us and has the rest of the development of our pipeline, as well as a partnership that we did in 2019 with Amgen to collaborate on the co-discovery of therapeutic antibodies to challenging target proteins. At present, the company has close to 50 and staff, split between Singapore and the US. We've raised about 60 million in financing, about 35 million in equity financing, with quite significant non-dilutive financing support from a number of partners and other sources of non-dilutive funding. The technology platform that I alluded to just now really allows us to pursue antibody discovery in a very different way to classical approaches, which really tend to give great antibodies to the immunodominant regions of a protein target, but not necessarily to the functional region that you're interested in. And so if you're lucky, that functional region overlaps with an immunodominant region, and your life is quite easy. But what we see in many cases, and particularly these hard targets, the functional region can be quite distant from the region that most antibodies bind to. And we believe that one of the reasons it's been so difficult to get good drugs to some of the targets of interest. To overcome this, develop this technology platform, which really takes full advantage of both the computational structural biology tools, modeling tools that are available now to infer what are those most likely important functional regions of a protein to target, and then through modulation and engineering of an immune response allows us to get antibodies that bind to that region specifically, rather than to the more kind of classic low-hanging fruit immunodominant regions. And this has turned out to be an extremely effective way to get highly differentiated functional antibodies. Turning to our pipeline, I mentioned in the introduction, we started the company really focusing on HER3, and it's been very exciting for us to see this program progress. We have a partnership with Cancer Research UK to collaborate on this target, and the program has just completed the GLP tox and GMP manufacturing, and we'll be filing the IND in the next couple of months. A second program which has come out has been focused on VISTA, so a very intriguing immune checkpoint target that we believe has very significant potential given all of the underlying data around the target biology, but which again really does need the correct therapeutic strategy in order to unlock the value that we're interested in. And behind that, we have numerous other programs that have come from the platform. We are very excited about a multiple myeloma program. Again, a highly differentiated approach that's really only been possible because of the platform technology, as well as a lupus antibody that we believe offers a very distinct novel therapeutic strategy for treating lupus patients. Talking briefly about HER3 and what the challenge of HER3 was, I think that there's been interest in this target for 10, 15 years, and a lot of appreciation of the role of HER3 in cancer and its activation of the PI3K pathway. And early attempts to drug HER3 really focused on trying to inhibit all of the ligand. But over the last few years, we've seen increased understanding of the role of ligand-independent activation due to high levels of expression of the dimer partner, TFR or HER2. And so when you have high levels really of any of these proteins on the cell surface, you end up with the formation of these signaling complexes. And so our hypothesis was that instead of just blocking the NRG1 axis, it was essential to inhibit all of these interactions. Turning to the structure, it's helpful perhaps to understand what this really means and why other antibodies weren't able to do this. So the first generation of HER3 antibodies are the what we call the classic ligand antibodies that bound to this relatively immunodominant region of the HER3 protein And as I said, it's a very effective way of blocking tangles. But when you have high levels of EGFR or HER2 on the cell surface, that process I described, or independent activation, will occur. And so a better strategy would be an alternative way of blocking the activity. And in our view, the best way to do that was an antibody that would block the dimerization interface. This is a great hypothesis, but with classical antibody discovery technologies, it's very, very difficult to achieve such an antibody. And that's really what led to several years of work in our platform technology development, was to find a way to really first of all identify specifically what a good epitope would be, and then to get antibodies to that epitope. The proof, as I say, is in the data. So what we see here is some of the data that we published here at Cancer Therapeutics, showing the ability of our antibody to fully inhibit the form, in this case of 2-3 heterozygous, and you can see that that's something that happens with the other versions of HER3 antibodies. This translates to a profoundly different efficacy profile, both in vitro as well as in vivo, in model after model, really capture the efficiency of HER3 inhibition and its ability to shut down PI3K pathway signaling activity. Over the last 12 months, as we put the program through development, we've also been building out the clinical strategy. We have several opportunities that we're very excited to pursue, including NRG1 fusions, which have gained attention recently, as well as castrate-resistant prostate cancer and cetuximab-resistant cancers, including colorectal cancer. And beyond these tumors, we believe there's very significant potential to expand. However, we think that there's a very clear line of sight concepts in these indications that I mentioned, and that's where our initial focus will be. So these trials will be starting in the next few months. To do, I'll turn very quickly to our second program. It's a VISTA antibody. An important immune checkpoint, increasing data showing the high levels of VISTA expression in tumors, particularly in triple-negative breast cancer and non-small cell lung cancer. There's been abundant studies showing the importance of VISTA and its ability to inhibit T-cell activity. With that, we use the platform to target a specific functional interface on VISTA, taking advantage of the information of how VISTA, which is one of the 7 class of proteins, interacts with ligands. And this allows us to identify a critical region to block and enable development of a neutralizing rather than a depleting VISTA antibody. Putting this in clinical development, we see an excellent in vitro efficacy profile, very comparable to what you see with PD-L1 inhibition, which is a great reassurance, as well as a remodeling of the microenvironment with a very profound expansion of myeloid suppressor cells, which is very much the phenotype that you would want to see with this axis. Additionally, one of the advantages of our program is that we have been able to target a conserved epitope in rodents, and this allowed us to full use of efficacy and tox models, unlike other programs which are generally restricted to using surrogates. And we see really potent monotherapy activity in triple-negative breast and colorectal cancer, as well as a very intriguing combination data with PD-1, which really ties back to some of the underlying data around the expansion of VISTA-positive cells as a response to PD-1 therapy. I won't go into the clinical strategy here, but suffice to say that I think it's a great program really focusing on indications where there's very compelling evidence both of the role of VISTA and its ligands. It's a very similar stage of development to our HAD3 program, with the manufacturing complete, GLP tox complete, and we'll be initiating phase 1 studies again in the next few months. Just a very quick snapshot of the team. We're fortunate to have a great team and an even better scientific advisory board, led by some of the experts in the field. And we believe we're very poised with an exciting year ahead, seeing these programs go into the clinic with a clear line of sight to clinical proof of concept. There's an exciting pipeline of programs coming up behind it. So with that, I'll close here. Thank you very much.
Thank you, Piers. That's an extraordinarily impressive pipeline that you all have developed in such a short time frame. And I wish you tremendous success in that regard. Hopefully, the audio has improved a little bit through the Zoom as well, with some people on YouTube. Am I coming through okay to you, Piers?
Yes, it's quite clear now. Thank you.
Okay, good, excellent. I think that we should keep moving at the moment. So I'm going to hand us off next to Bradley Pride, who's the co-founder and COO of 13 Biotech. If you could please go ahead, Brad. Thank you.
How's my audio? I'll take it from anybody.
I think you're coming pretty well.
Fantastic. So I'll go ahead and share screen here, jump right in. So there's no secret that the failure rate in drug discovery is about 95%, which makes it difficult for investors to invest in the space. And in fact, there's actually a decrease in biotech investors' activity over the past several years. And we think from both a passionate basis and in just kind of a pragmatic basis, that doesn't have to be the case, because especially when you're dealing with fields such as oncology that has been around for a very long time, there is just myriads of different data points that can be analyzed. But in order to do so at level and the magnitude of data that's available, you have to do it intelligently. So what we're doing is implementing biology-driven AI into the drug discovery process. First, let me introduce myself and my co-founders who are not on, and then I'll jump into some of our success stories and the methodology that we follow to have those success stories. So the first person I'd love to introduce, Neil. He's a Forbes 30 under 30, and him and alongside Olivier to his right here are two of the first members of the Precision Medicine Institute at Weill Cornell, which is now a 300-person lab and the largest precision medicine institute in America. And then there's Corey, who is our chief data scientist, and she has a background in applied ML. She was actually the fastest graduate from a PhD program in Cornell history, and was formerly at a very large AI hedge fund called WorldQuant. Now, my own background, as you can see from the imagery here, I don't have a PhD, so by default, I would take the title stupid co-founder. But my background is different in that I had started off in finance in London and moved into trading, and had an important event which really spurred my curiosity and motivation into this field, which was the passing of a family member and relative. So I moved into tech. So my first business as the CEO, and then came back to America and started applying my experience across a multitude of different industries. It was when I met these, fantastic scientists that I realized that there's a crossover in my experience in ML and AI, and a real impact that could be made in the fight against cancer and drug discovery. So just to take a little bit, a step further back into the kind of 13 Biotech and the successes that we've had along the way, it would start back in 2011. In 2011, we began as an idea in Weill Cornell's Precision Medicine Institute, which I mentioned earlier. And in this setting, we were able to build out technology and databases alongside real-world results so that we were changing our methodologies and building out our IP in a lab setting. So really understanding the implementations that our predictions were having. So as we moved towards 2015, we started to branch out and wanting to test out our theories, our hypothesis, our predictions in industry, and partnered with our first big pharma partner to validate an adverse event prediction that we had. And off the back of that, we're noted in cell chemical biology. Moving into 2016, we started to kind of develop out our commercial structure. And we did that first with Oncoceutics, who were recently acquired by Chimerix, and partnered with them to identify their mechanism action and lead indications. And off the back of that, in the same year, got our first funding round from RDB, which is a part of General Catalyst. In 2018, with AstraZeneca, we validated our combination synergy prediction module. And we're also featured in Nature Communications for the work that we had done with Oncoceutics. So what we had done with Oncoceutics, we came in with them when they were in a phase 2 trial, and they're testing their lead indication against 6 different cancer types and not really having the level of efficacy that they wanted. So what we're able to do is determine the mechanism of action, which they didn't know because they found their asset in a phenotypic screen. And then we were able to identify the mechanism of action, off the back of that, make a prediction on what the lead indication should be. And within this lead indication, which we found to be a methylated glioblastoma, they restructured their phase 2 trials and had great success to the extent that patients who were, given a very short time period to live are now, thriving. This is a case study I can actually jump into later. So 2020 for us, based off of the successes we had seen, was all about signing up new partnerships with pharmaceutical companies. And it started off with a fundraise that we had from Primary Meridian Capital at the very beginning of the year, which was fortuitous timing considering that, coronavirus broke out and really disturbed the industry in some aspects. And we've been just shaping our business model to work with these different pharmaceutical companies and being able to announce them along the way, as well as winning some awards. So we were awarded the most innovative technology at China America Innovation Summit. We were the winner of the Reuters Pharma Entrepreneur Award. And so far, we've been able to legally announce the partnership with Jubilant, where we identified biomarkers in optimal patients. And we have many more partnerships to have announced as we rolled them out in 2021 and finish contracts, but also get the permission to go ahead and talk about them. I think the next thing to go over is really explaining what methodologies do we employ, what's our secret sauce, so to speak, to give us the results that we've been able to have and be clinically validated in a variety of different ways. The first thing that is by far one of the most important things alongside the actual IP of machine learning and having accurate predictions is it starts really with the data. So we have out of the over 10 years of research and development, 2 years alone were just spent on building out the infrastructure for the data engine that we have and employ against our different types of analysis. And on that database, we have over 40 curated different data types from over 160 different data sources, from public, private, with billions of data points spanning the biological, chemical, and clinical domains. Now, if you look to the left, the infographic here, what we're trying to kind of depict is the fact that we have 3 different kind of main data sources. Proprietary data, so that's coming from academic institutions and then pharmaceutical partnerships that we've established previously. And then public domain data, which would be per volume one of the largest data sources that we have. So the value add that we really bring there is, if you think about a very, I would say an oversimplification of what we can do there, it's kind of think about Google. The World Wide Web is a vast tool of tons and tons of different information, but you didn't have Google's algorithms to really reach out and then automatically absorb that information and to decode it in a way that which is understandable to you. It wouldn't be very useful because it's just too far, too vast. That's what we built out. The data engineers have built out ETL pipelines, which automatically scrape the world's publicly accessible databases for a variety of different data types. But then it goes a step beyond that, and there's a scientific exercise where the data engineer and the computational biologist and machine learning scientists within our company are working together to assign relevancy scores and confidence scores based on the particular type of product we're looking to solve. And then with any new partnership that we do establish, we do bring in their data to determine its relevance, to determine the confidence we have, and see how that can help solve the problem in particular that we'll be looking at. So to the far right there, what I'm exemplifying is just the different types of data that are existent within our database that we've pulled together from the live publicly available sources that have been filtered through our systems, but then also from proprietary partnerships that we have as well. So thinking about what problems we tackle. So when we were going back and we were creating this back in 2011, we're thinking the way to be a true partner to a pharmaceutical company and really help advance drugs through different phases in the drug discovery pipeline. It's important to not only incorporate all the different data types in order to be able to do so, but then tackle the key problems that pharmaceutical companies come across. And in thinking about the key problems and then trying to categorize the different areas in which we tackle problems, we came up with this tribunal where we're looking at linking treatments to patients, patients to targets, and targets to treatments. So you can see we're not really seeing it a linear function. We can kind of come in and insert ourselves as a partner at any kind of stage within drug discovery. So you may be looking to link a treatment to a patient. We're looking at biomarker identification, looking at drug repositioning, potentially drug combination synergy to really add to your portfolio and generate new IP. We're looking to link patients to targets through determining gene essentiality and genetic interactions, or targets to treatments that determine the target deconvolution and drug toxicity. But laid it out here in a format which really allows anybody, and happy to share these slides. I'm sure it'll be available through the organizers of the event. But this gives an idea of the module, breakdown of what it does, where it isn't very self-explanatory. And then there's a link to either a case study where we had worked with a pharmaceutical company to prove out within the clinic, or where we've been heavily scrutinized by one of the most reputable peer-reviewed journals that exist within the world. So that's the science. That's what we have been doing. And to think a little bit more about the business model and how we work with pharmaceutical partnerships. So what we typically do is we take more of an approach of, I'd say, an outsourced research and development team. So we form research and development collaborations with our pharmaceutical partnerships, because every project that we embark on is very much a scientific exercise. We do have 14 patented algorithms that fit into those 7 different modules that I just went over. And we do have the database, but as this data comes in, we're forming that for the project, and we're rerunning our methodologies with new data so that it's never a plug and chug software as a service type situation, and not the business model we employ. And as an output of the work that we pursue, we give what we call actionable mechanistic insights. And that's a report of a ranked list of different, given an example of oncoceutics, a ranked list of different mechanisms of actions ranked by efficacy and toxicity. We'll then give that to the partner, where we aim to marry our objective as well as subjective analysis with their domain specific expertise. And as they bring that actionable insights into the clinic, we always stand by our partners in a continued effort as new data becomes available. What I'll do now is I'll jump into one of the case studies, which helps kind of put this into a little bit more of a perspective. So the success story that I kind of picked to go over here is in regards to oncoceutics, just because I think it has that patient, you can put a face to the name of how that's affecting people in the real world, but also because oncoceutics was recently acquired. They had 2 lead assets, one of which was the one we worked on for ONC201, and they're acquired for considerations of over 400 million. So I think that goes to show that although we're not holistically responsible for all of their success there, we did have a good part in generating the IP that has helped them get along the way. So going back to this case study, I know I briefly went over it before, but when we encountered oncoceutics, the background was that they had actually discovered ONC201 through a phenotypic screen for the P53 integrated stress response. And they were stuck, as I was mentioning, in a basket phase 2 trials because the anti-cancer mechanism was unknown. And this blocked downstream positioning to specific biomarkers. So our objective was to identify the novel binding targets for ONC201 and pinpoint specific patient/indications for future trials. And the core modules that we used in that area where I outlined before was the target deconvolution, the gene essentiality, and biomarker identification. So first job was to identify ONC201 as a selective DRD2 antagonist, so basically finding the mechanism of action. So in step and phase from a very high level, the first step here was using the OTB, that's the 1,3-bio target deconvolution module. We ranked all possible targets and the mechanism of action for ONC201. The input data sets actually wasn't extremely advanced there. We're actually able to heavily rely on our database as well as being, in part supplemented from the data that they provided. They gave us the chemical structure of ONC201, efficacy screening against 60 cancer cell lines, and post-treatment gene expression changes. So the rest was our database. So we top ranked predictions for ONC201 at MOA was selective antagonism against DRD2, which is a novel anti-cancer target. This was brand new science at this point in time. And a DRD2 binding prediction was validated through preclinical assays and clinical readouts. We then moved on to the next phase, was pinpointing, what exactly was their indication based off this new science, which we discovered with them. So the first thing that we went after was using the OTB gene essentiality model. We were able to rank how susceptible 300 different cancer subtypes would be to the inhibition of DRD2. And based off this ranking, we identified 2 indications actually as very likely based on the probability scoring that we had. So one was glioblastoma, another was pheochromocytoma. These look like ideal areas for clinical development. And specifically, we found that H3K27 methylation was identified as a predictive biomarker for ONC201 treatment in glioblastoma. So this is where we use the biomarker identification. And we validated this prediction, then worked with Oncoceutics to start a new phase 2 trials in H3K27 methylated glioblastoma. So obviously the question being, how did that go? Phenomenally. And I think when, it's great to go into the science because it's absolutely essential, but when you do actually have an effect on the patients, it's amazing to put a face to the name there. As you can see here in the different pictures I put here, there's very clear recessions and tumors within the patient's brain. And I think as, business people and people from a scientific background, we don't really expect to be in People magazine, but I think it is fantastic to see those results where we're seeing people who have, father of 4 and, mothers who'd been coming into these trials who'd been giving a week to live or a month to live, and they're now stable, and they have been stable for over 18 months now when these trials took place. So from a business perspective, ONC201 is now moving into phase 3 trials. The company was recently acquired. And here's a quote directly from the Chief Scientific Officer, Joshua Allen, where he says that 1.3's platform helped solve problems that we'd been working on for years and potentially save several years of additional work. And I expect this approach will be integral to the future of drug discovery, and us from a very biased point of view would agree. And then just looking at, time saved. So it's gone on for 4 years without success using traditional techniques to find the novel science that we were able to find. And in 3 months of work, we were able to validate these insights when working alongside them. So that is the end to my presentation, but would love to open it up for questions from anybody who would like to ask any.
Brad, that was terrific. Thank you. A couple of follow-up questions that have sort of been passed along that might be interesting. So, it tends to orient a little bit towards your commercialization and your business model, which you spoke about. So 2 questions. One, do you have any intention as part of strategy near term or perhaps midterm to get into drug discovery for your own benefit? And start to build out a pipeline, a clinical pipeline of your own. One, and for the near-term opportunities that you are currently exploring, do you have, are you all able to, or have you been successful in some situations with getting the ability to participate in downstream economics from your partners' therapies that you are helping to solve their problems or maximize their opportunity for success?
Yeah, great questions, and I can answer both in one answer. So I think in consideration of whether or not we're going to go and pursue our own, asset development and actually, develop out pharmaceuticals, we'd take a look at the current business model / models that we're pursuing now. So there are 2 different kinds of business models. So one is there's certain types of modules which we can employ that have the potential to be very scalable. And that way we could work to more for fee for service. So things like toxicity predictions very much could have our business model turn into a software as a service type provider. However, for the most part, where we're generating real genuine IP, like working with Oncoceutics. We are actually taking an approach of securing royalties and as well as milestone payments. So we'll do an upfront fee that will cover the cost of doing the work, not really making a profit on there. And then the rest will be, either optional for the results of our studies for our partner to take further downstream. And then if and when they do, that's when we recognize the payments, with the thought process being, if we provide such good backing for novel science, then of course they would want to. So it's a great way to actually develop out a business model in that partnership format. So yes, we have been able to secure those downstream payments and royalties with the partners that we've been working with, and have several discussions for some that we're very excited about, in particular drug repurposing as an opportunity, and then combination synergies, which is really kind of fascinating IP generation. And thinking about how does that business model lend itself to potentially become, an actual pharmaceutical company itself, it does quite well. And I think that's the kind of downstream attraction for investors for what we're doing, because there's two different ways to kind of look at that. It's the reason that you're losing interest in this particular, biotech investors is because of the failure rate. It's really great to say, hey, if we made it, we can make $1 billion, but there's a 5% chance of that. So the standard deviation from your original investment is extremely huge. Now we can go and we can sign, a lot of different partnerships where we get a piece of that 5% possibility. But what is really attractive to investors is the fact that we can say, not only are we able to capture value here, we're also able to decrease that failure rate and capture value further down by actually developing out our pharmaceuticals and then kind of engaging with pharmaceutical companies when we've already pushed something through preclinical trials.
That's terrific. It sounds like, it's a very exciting opportunity, both with what you're doing in the near term and, for potentially building out, additional strategies and business success stories, but also, an evolving business model for 1-3 itself. So congrats to you. I'd love to talk to you more about data later, but we'll have to do that in a separate conversation.
Sounds like a plan.
So we've run out of time so we should move on to our next panelist. And as we mentioned, we're not going to be able to hear from Prestige at the moment. So I'm going to turn over to Charles, and I hope I'm pronouncing your name correctly, Charles, but Charles Thor, who is the CEO of TRACON.
Hi, Joshua. Yeah, you're pronouncing it correctly. Can you hear me okay?
Yeah. Fortunately, they seem to have solved the audio problem, so it's all coming in quite smoothly now.
Great. Well, pleasure to be here. Pleasure to meet you, and a pleasure to present TRACON. So I'll get started. TRACON's a publicly traded NASDAQ company since 2015, based in San Diego. I'll make some forward-looking statements throughout the presentation. So today I thought I would briefly discuss 3 of TRACON's key value propositions. One is our lead asset, which is a best-in-class checkpoint inhibitor called envafolimab. That's in a pivotal trial in sarcoma with expected data this year, final data next year, and potential approval in just 2 years. Next is our broader pipeline that includes 2 additional clinical stage assets. One is TRC102, which is an inhibitor of the DNA-based excision repair pathway. That's in a broad phase 2 program in collaboration with the National Cancer Institute. And also we have a phase 1 CD73 antibody called TJ4309 that's in a phase 1 trial in combination with Tecentriq, where we also expect data this year. One of the key value propositions of TRACON that's quite unique is our business development engine. We call it our product development platform. It's a platform of CRO independent clinical development and commercialization expertise that allows us to work with ex-US partners who are looking to optimize the value of their product in the US. Through our platform and our ability to develop drugs quickly and then take them all the way through commercialization and share profits with our partners, we've been able to execute 3 important collaborations with 3 separate Chinese companies that I'll discuss. One is a partnership with Alphamab Oncology and 3D Medicines around envafolimab, and second is partnerships around the TJ4309 asset with IMAB, as well as earlier stage assets. Importantly, we have capacity and appetite for more deals now, and I'll emphasize as part of the presentation that we're looking for first or best-in-class assets from Asia for companies who want to develop them quickly in the US, have a partner that can also commercialize that drug, and also share in the profits. We have a very low burn rate given we're CRO independent and we do trials at about half the cost of most companies that are beholden to CROs, and our current capital provides about 20 months of runway into very late 2022, which is well past both the interim data expected this year and the final data for our pivotal trials asset, which is envafolimab. So let's talk a little bit about envafolimab in more detail, as it's our most late-stage asset and represents our near-term commercial opportunity. What's special about envafolimab, it's a checkpoint inhibitor. It inhibits the PD-L1 pathway. It's given subcutaneously, and that's what makes it special. It's given as a simple, rapid, subcutaneous injection, very similar to how you would receive a flu shot, and that's a paradigm shift. We've been rapidly executing on developing this asset since licensing in late December 2019. And currently we're already dosing in a pivotal study following a very successful FDA meeting earlier in 2020. Our focus is an unmet need orphan indication in sarcoma. It's an indication that can give us significant revenues, $300 million estimated initially. And then as we grow the franchise in other sarcoma subtypes, potentially increasing that sales figure substantially. Importantly, we picked sarcoma because it represents a fast to market strategy. It's a very high unmet need indication. There are no other checkpoint inhibitors being studied in pivotal trials. And by delivering interim data this year and potentially achieving breakthrough status, final data next year, we hope to commercialize the drug in 2023. The other important thing is by leveraging our product development platform of CRO independent research, we can do the trial at very low cost, less than $20 million. And we offer our partners significant financial upside. The royalty burden tops out at what equates to a profit share for our partner. So instead of our partner getting, say, a 10% royalty from a big pharma company, they will achieve a profit share roughly that's potentially 3 or 4 times the royalty they would achieve from a standard big pharma license. Our second highlight is beyond envafolimab. So envafolimab, clearly our lead asset in a pivotal study licensed from 2 Chinese companies, 3D Medicines and AlphaMab Oncology. Our second asset, TRC102, we work in collaboration with the National Cancer Institute. That's in phase 2. That's an inhibitor of DNA repair. That's available for global license. And then TJ4309 is our phase 1 CD73 antibody that was licensed from another Chinese company, I-Mab Biopharma, that's also in phase 1. Another deal with I-Mab included access to a broad bispecific pipeline. I want to highlight a little bit further our partnering platform. It's such an important part of our business and such a value add for partners who are looking to optimize the value of their asset in the US. What we do is everything needed to execute a clinical trial, all the regulatory interactions with the US FDA, and the commercialization of a first-in-class or best-in-class asset. We're looking for assets that have clinical data or are IND ready. If we find an asset we like and we work with a partner, what we generally do is pay for all the clinical trials. And then we would share the cost of commercialization and importantly, share the profits with our partner from US sales. That's a much better deal typically than what most pharma companies will offer a potential partner in Asia for a first-in-class or best-in-class asset. We have leveraged that product development platform to execute 4 important partnerships. Most important is the envafolimab partnership with 3D Medicines and AlphaMab Oncology. That was done in December 2019. In 2016, we validated our platform through a partnership with Johnson & Johnson around a prostate cancer asset that's currently phase 3 ready. That's also available for global license. And in 2018, we executed 2 deals with I-Mab, one around the CD73 antibody now in phase 1, and the other around access to a bispecific antibody pipeline. Importantly, while we are experts in oncology, we're interested in assets that potentially are developed in any therapeutic area where marketing would be done to a physician specialty audience. And as I mentioned, we can license assets at any stage of clinical development or assets that are IND ready. Let's talk a little bit more about envafolimab and understand what makes it so special. As everyone I think well knows, checkpoint inhibitors have revolutionized the care of cancer patients, and there are now 6 approved in the US. Each of those is given intravenously. Disadvantages are patients need to go to an infusion center, get pre-meds, get an IV, sit in a chair for at least half an hour. They risk an infusion reaction, which requires potentially longer time in the chair and additional complications. Contrast that to envafolimab. It's given in less than 2 cc's in 30 seconds, sub-Q, much like you would receive a flu shot. It can be done in a doctor's office. It's an injection, not an infusion, so you don't have the risk of infusion reactions. It's fast and it's easy. It's a paradigm shift. It's as simply given as shown in this picture. Literally, this is the back of the arm of a patient dosed with envafolimab, a 25 gauge needle inserted under sterilized skin. 30 seconds later, the injection is complete and the patient may go home. Again, very different than an infusion center chair required for an IV checkpoint inhibitor. That's what gives it a best-in-class profile. Importantly, evidence indicates that envafolimab is as active as reference checkpoint inhibitors like Opdivo and Keytruda. When you look at clinical trial data from a very similar population of patients, these are all patients with MSI-high colorectal cancer that failed 3 standard chemotherapies. The response rate for Enva, 32%, nearly identical to that of Opdivo and Keytruda. Importantly, our partners in China, 3D Medicines and Alphamab Oncology, have now filed this drug for approval in China based on the data in MSI-high cancer. Recently, that application received priority review status by the Chinese NMPA. With respect to our approach, we recognize the fact that checkpoint inhibitors can be approved by the US FDA in unmet need indications based on objective response rate. And if the unmet need is severe, the objective response rate needed to approve checkpoint inhibitors in refractory settings is fairly low. For example, Keytruda was approved based on a 13% response rate in gastric cancer. Tecentriq was approved based on a 15% response rate in bladder cancer. And Opdivo was approved based on a 12% response rate in small cell lung cancer. The reason being, the standard of care for those diseases was so poor at the time the checkpoint inhibitors data was made available. We know for sure that drugs can be approved in sarcoma based on fairly low response rates. In 2020, tazemetostat was approved in epithelial sarcoma based on 2 small trials, one with an 11% and one with a 15% response rate. Unfortunately, for patients that have certain sarcomas, the unmet need is severe. We're focused on 2 sarcoma subtypes called UPS and MFS. They account for approximately 20% of all sarcoma. Like most sarcomas, they're treated first line with chemotherapy with an expected response rate of about 17%. Unfortunately, when patients fail first line therapy, they have one approved therapy for their disease. It's called Votrient, and it has a 4% response rate per the package insert. You heard me correctly. Patients with refractory UPS and MFS have one approved therapy with a 4% response rate. Now, given those data, there are transformational data recently published with checkpoint inhibitors in these same patients. Specifically, at ASCO 2019, Keytruda demonstrated a 23% response rate in refractory UPS and MFS patients. Last year at ASCO, further data showed that dual checkpoint inhibition with Opdivo and Yervoy generated a 29% in refractory UPS and MFS. So just imagine that. If we can approve a checkpoint inhibitor in refractory UPS and MFS and mirror the data with Keytruda as a single agent or with Opdivo and Yervoy as a combination and generate response rates as high as 30%, that would be a transformational standard of care change compared to the only available approved therapy, Votrient, with a 4% response rate. Despite these encouraging data, there's no company focused on approving a checkpoint inhibitor in sarcoma. And our pivotal study is the only study moving forward to enable approval of a checkpoint inhibitor for these patients. The other clear advantage is not just meeting the unmet need, but doing it with envafolimab and representing the most convenient checkpoint inhibitor by virtue of its subcutaneous dosing, and as an approved therapy that would facilitate reimbursement and physician and patient access. Let's take a quick look at the timeline that has resulted in us now dosing in the pivotal study. We licensed the drug in late December 2019. Our partners fortunately had completed phase 1 testing in the US and had dosed over 700 patients globally in the US, Japan, and in China. Leveraging that data, we met with the FDA in May and secured concurrence on a pivotal trial design whereby we study envafolimab as a single agent in cohort A and in combination with Yervoy in cohort B. Our goal is to mirror in cohort A the data with Keytruda of about a 15 to 20% response rate. Our goal in cohort B is to mirror the data with Yervoy and Opdivo of about a 30% response rate. If we're able to achieve that target product profile, we expect to approve the drug both as a single agent and in combination with Yervoy. Since the FDA meeting in May last year, we submitted the protocol to the agency. It was cleared in August, and we initiated dosing in December. We expect interim data the second half of this year, final data next year, and expect to be marketing the drug in 2023, assuming we achieve the target product profile I've just discussed. I would like to emphasize one thing about the clinical trial, and that is the bar for a positive trial is not high. For example, we take refractory UPS and MFS patients and they're enrolled into either cohort A of single agent envafolimab treatment or cohort B of envafolimab plus Yervoy. In each cohort, the goal is to show a response rate that's statistically superior to the known 4% response rate of Votrient. So in other words, cohort A and cohort B are not compared against each other. They're both compared to the historical 4% response rate of Votrient. In other words, we need 9 of 80 responses in either cohort or an 11.25% response rate to have a positive trial. Clearly, those responses need to be durable in the majority of cases. Excuse me. In terms of the initial market in refractory UPS and MFS, as well as other sarcoma subtypes known to respond to either single agent checkpoint inhibition or dual checkpoint inhibition, the initial market is projected to be at $300 million. But importantly, we don't intend to stop development in sarcoma with the first approval. We expect to go first line in combination with chemotherapy, much like other checkpoint inhibitors have done in other indications. For instance, Keytruda, initially approved in refractory lung cancer, now approved first line. We will take that same opportunity to label expanded sarcoma and mirror the development of other checkpoint inhibitors to potentially increase the market opportunity. Let's briefly talk about our 2 other assets. TRC-102 is in a phase 2 studies, many studies in fact, that have been sponsored by the National Cancer Institute. Importantly, it was profiled last year in Cancer Cell, where the publication talked about an exceptional responder with colorectal cancer that responded with a near complete response for almost 4 years, treated with Temodar and TRC-102. Importantly, that was impressive because colorectal cancer generally doesn't respond to Temodar. What they discovered was the patient seemed very sensitive to TRC-102 and Temodar because of certain genetic mutations. In particular, they had a methylated MGMT promoter, meaning they didn't make MGMT. They also had 2 mutations in double-strand break repair pathways that rendered that repair pathway obsolete. That made them very dependent on base excision repair to repair Temodar-induced DNA damage. By inhibiting that pathway with TRC-102, we generated an impressive near-complete response. That hypothesis was further solidified by the fact that 11 more patients came onto trial. The 10 that had MGMT expression did not respond, but the patient who also had methylated MGMT promoter and did not express MGMT also responded to Temodar plus TRC-102. These data are consistent with previous data we have presented in refractory GBM, and also data in advanced localized non-small cell lung cancer that was presented at ASCO last year. Based on those data in this publication, we expect continued development of this drug in collaboration with the NCI, both in lung cancer and also in advanced localized non-small cell lung cancer. Let's talk about our 3rd clinical stage asset, the antibody TJ4309, which is a CD73 antibody, which inhibits the formation of adenosine at the tumor interface, adenosine being an immunosuppressive metabolite. By inhibiting its formation, TJ4309 is expected potentially Tecentriq, a model PD-L1 inhibitor. Currently, we're dosing in a phase 1 dose escalation study, and we expect to report data this year of safety, PK, and potential efficacy of TJ4309 in combination with Tecentriq, and then based on that, discuss further potential phase 2 development. This is an asset where we're partnered with I-Mab. I-Mab is another Chinese company. Our initial deal with I-Mab was 2018, and we're responsible for the initial clinical development of TJ4309 in the US. Importantly, as a sign of how quickly we work with partners, we filed the IND for TJ4309 less than a month after we initiated the license in 2018. I want to finish by just talking a little bit about, again, our partnering platform, because we're eager to license in additional assets from Asia, companies who want to maximize the value of their compound. We are a CRO independent group. By being CRO independent, we don't pay others to run trials inefficiently. We run the trials ourselves at lower cost, faster, and at higher quality than a CRO. We leverage that and use that improved efficiency to help our partners move this forward very quickly. As evidenced by envafolimab, we did a license for envafolimab in December 19. We have now entered and started dosing a pivotal trial in December of 2020. That's less than a year. We expect to complete it this year. That gives you an idea of the pace of development using the TRACON product platform. The way we make it work for our partners is the following. We engage in a cost, risk, and profit share aligned structure. So in other words, if our partners discovered a molecule and invested in that, their manufacturing molecule and invested in that, we'll take on the responsibilities of regulatory and the clinical trials. Once the drug is approved, we will then share the commercial costs and we'll share the profits with our partner. What does that mean? Well, at an 80% profit margin, what that means is our partner recoups about 40% effective royalty. That's about 4 times the royalty they may typically receive from a big pharma company for the license of their asset. So companies who believe in our assets and want the big economics on the back end are ideal partners for TRACON. Importantly, we file a BLA or NDA in the United States. We provide that dossier to our partner. They can use that to file in their regional territory, gain further approval, and they would own those rights completely. We're focused on oncology, but we can do trials across multiple therapeutic areas. Areas where we see an unmet need, we'll be eager to explore. We've been recognized for our clinical trial excellence, and clearly our partners have benefited from partnering with TRACON. With respect to alpha-mab oncology and 3D medicines, initiation of a pivotal study, I-mab rapid filing of their IND. J&J, we did a phase 1 and phase 2 trial in 3 years for them, and we're eager to acquire more assets at the present time. So in summary, TRACON, clinical stage company, multiple assets, including a pivotal study around the lead asset and envafolimab as a best-in-class checkpoint inhibitor, with the ability and the eagerness to do additional research with companies for first or best-in-class assets and optimize the value of those assets in the state. Appreciate your time and welcome questions.
Thanks very much, Ross. That was very impressive as well. And the PD-1 checkpoint inhibitor cutaneous application is quite revolutionary. It's incredible. You have to find some combination therapies that can utilize the same injection method. I think in time we're going to move.
Sure.
Okay.
Great issue. Thank you.
Okay. I want to move ahead to our next presenter, Trio Pharmaceuticals, which will be by Shimmick, who's the founder and CEO. Share, are you there?
Can you hear me? Everyone hear me?
Yes, we can hear
Hello?
you. Can you?
But I cannot see the, it says that I cannot start my video. The host had stopped it. Hello?
Yes. We are you able to start your video?
I can't start the video because the host had stopped it. That's what they're saying. Okay, I guess then I can start with sharing the screen.
There you
Let
go.
me start the video now. Can everyone see me?
And you're on video too. Yep.
Great. So just, all right. And can everyone see the screen?
Yes.
Hello? Okay, great. All right, so thank you for everyone for and and hello to everyone and hasayo to everyone in Korea, and wanted to say thanks for giving me the opportunity to present today about what we are doing. So we are a cancer therapeutics company, and coincidentally, like our speaker, we are also based in San Diego. And before we start digging more into our technology, I just wanted to give you a brief idea who we are and what our team members have accomplished throughout the years. So we are a very strong cancer drug development team. People who have worked in the cancer drug development for more than 30 years are now currently working at Trio. So Alan, Grant, they previously had worked at Seattle Genetics and were very crucial in the development of the first ADC drug that came up from Seattle Genetics, and that's the Etcetris program. And then Bill Brady has a strong background in antibody therapeutic design, invented and developed the drug Orencia while I was at Bristol Myers. My background has been in the antibody cancer drug development. We all collaborated and worked together previously at a company called Ambrx, and which antibody drug conjugate site-specific conjugation technology company. And as we were working on, I think we were all the time coming up with this idea how to make cancer drugs more and more tolerable rather than making more efficacious. And I think our mission is actually in the name. It's actually that the fact that the cancer drugs are failing for the patients in the clinic, it's not because lack of efficacy, it's because of lack of tolerability. And that's why, or rather because higher toxicity. And that's why the name is actually toxicity reduction in oncology. And to tell you what we are doing is actually we have invented the first-in-class dual action antibody drug conjugate. So as you are aware, currently there has been in the 2020, since April 2020, there has been a lot of ADC deals that has happened. And as you would remember, that all those ADCs are single action and they are only doing tumor growth by eliminating cancer cells. In our case, we are doing two actions. We are stopping both tumor growth as well as stopping immunosuppression at the same time. Our antibody drug conjugate has two arms, two targeting arms rather. One is the blue one shown over here. It can recognizes the cancer cell and eliminates the cancer cell by receptor-mediated internalization toxin, and that's how it stops the tumor growth. Another arm shown over here by orange recognizes the immunosuppressor cell, internalizes the toxin by receptor-mediated internalization, and eliminates the immunosuppressor cell to stop the immunosuppression. And that's how it actually stops both tumor growth as well as immunosuppression at the same time. And the benefit of our approach is that in an IO treatment landscape, where the treatment is actually dependent on prevalent T cells in the tumor microenvironment, we are creating more immunogenic tumor microenvironment. And then these two diagrams parallelly can shows that current ADC drugs or current cancer drugs, they're only stop, since they're targeting only cancer cells, they're able to stop the immunosuppression effect from the cancer cell, whereas the immunosuppressive effect from immunosuppressor cells remains intact. And that's why it doesn't break the immunosuppressive circuit. And in contrast, on the right-hand side, you see that because of our drug, the immunosuppressive circuit is entirely broken down. The suppressive effects due to the cancer cells, as well as the immunosuppressor cells, is entirely broken, and it will create a more immunogenic tumor microenvironment, whereby the effector cells would be more strong, effective immunogenic attack on cancer cell to enhance any kind of cancer-free survival by eliminating cancer cells. And our approach, only way to simultaneously stop tumor growth as well as selectively stop absorption in the tumor microenvironment. Current technologies actually are not able to do that. They are able to do that by combo, and when you approach the combo, it leads to stopping immunosuppression outside of the tumor microenvironment, and that leads to a lot of immune-related adverse events. Our platform is that we can actually generate novel drugs from existing drugs. We can stay on the fag end of the discovery, but our approach is actually to go fast towards the clinic. And what we are doing is, because we are bringing in the dual functionality, that's the novelty that we are bringing in, we can technically validated binders for which we know already PK/PD and safety profile is known. And we can combine them for targeting two different aspects, stopping tumor growth and stopping immunosuppression. And that's why our platform has both, in a way, is a first-in-class, best-in-class, as well as fast-follow approach. We have claimed varied receptors that are associated with tumor growth, as well as immunosuppression. And that's how we have actually built our platform. We have established the novelty, and one is actually doing what we are doing. And we have also, for a plug-and-play modular development, we actually claimed, or rather come up with a very stable molecular framework where we can graft different binders onto that framework. And this entire technology is wholly owned by Trio Pharmaceuticals. I also wanted to point out that since right now we are working with clinically validated binders and targets, our platform is also amenable with novel targets and binders. So as I pointed out, we have right now a best first-class pipeline, reduced development risk. We are using clinically validated binders and toxins, and we can get, based on approach, both first to third line approval. And we are working on drugs for triple-negative breast cancer and small cell and metastatic prostate resistant prostate cancer, cancers that are of high unmet medical need. We have identified a lead program that we want to take it towards the clinic very fast. And here, right now, from our current time frame, we can get into IND candidate within 24 months. Other cancers and drugs that we are working on the pipeline, which is only 6 months behind us. Our lead program, if you wanted to brief what it does is basically, it's eliminate cancer cell and myeloid-derived suppressor cells. We have defined binding domains and cytotoxins from approved cancer drugs, designed in such a way that we have a high affinity towards the tumor antigen, such that the drug action would be restricted in the tumor microenvironment. And thereby we can actually accomplish PK/PD, bioavailability, and a safety profile driven by the tumor receptor rather than by the myeloid-derived suppressor cells receptor. We can also reduce the tumor toxicity, as I pointed out, by restricting the direction within the tumor microenvironment. We have looked through the patent. We have an established FTO for both the bindings and the cytotoxins. And wanted to point out why there is a clinical basis. MDSCs are actually associated with poor survival. And these are the two graphs that I wanted to point out, particularly for metastatic cancer, as well as non-small cell lung cancer. It has been shown that higher level of MDSCs actually leads to very, very low survival. And similarly, in non-small cell lung cancer also, higher level of MDSCs actually leads to very low survival of pair cancer patients. And we also wanted to point out that we have identified patients that would be benefiting from our approach. We went through the cancer genome atlas and we found that the receptors actually co-localize in the same tumor biopsy. And that's we identified that the MDSCs are actually penetrating into the tumor microenvironment and can actually create a very non-immunogenic environment. And that's why our approach would be working. In terms of accomplishments, within a short span of time that we have accomplished, we have shown that our molecule is very stable. It can be scaled. We are already getting very high expression titers, high yield, and we have done a stability assessment of the molecule. And it's actually stable, or very stable. We are getting less than 1.5% aggregate rate of time. And we have shown tumor selectivity, which is very critical for us. And we have shown there is a high binding affinity towards the tumor antigen than the MDSC antigen. And we have shown that there is in fact, significantly lower binding affinity towards MDSC antigen than in other approved drugs. And the same binding is retained to the tumor antigen as approved as a drug for the tumor antigen. We have also shown cell killing in vitro as well as in vivo. We have shown that there is a higher tumor killing of tumor cells than the MDSC-like cells. Similarly, in growth in vivo models, we are using tumor xenograft. We have shown that there is statistically significant growth delay of MDSC-like cell xenograft versus the tumor growth inhibition of the tumors. Just wanted to give you a brief idea with regard to our the data that we have accomplished so far. As I pointed out, our data looks like that the tumor antigen has a higher affinity. We are currently showing that less than sub-nanomolar binding affinity, and then double-digit or single-digit binding affinity towards the MDSC antigen. And that is very much reflected in the in vitro cells. As you can see over here in the lower panel, there is indeed a very high killing of the tumor cell killing and very low MDSC-like killing in the in vitro assay. And in the growth fusion of the cancer cell xenograft, we are indeed seeing a very statistically significant growth delay of the MDSC-like cell xenograft. And right now we are on track to doing right now our tumor cell line xenograft study. We're expecting to get the data soon, and we would be expecting a higher delay than compared to the MDSC xenograft. And wanted to point out again why we are a different class of molecule compared to us. In terms of ADCs, we are definitely advancing the ADC field. Currently, as you are aware, that first generation ADCs are primarily geared towards cytotoxin delivery. Second generation ADCs that have come right now, they are primarily activating T cells or myeloid cells rather. They are eliminating also cancer cells, but indirectly. In our approach again is eliminate both cancer cells as well as immunosuppression. So it's a tumor regressor as well as an anti-immunosuppressor. Again, I wanted to point out in terms of since we're making a bispecific antibody, we do not deliberately use the word bispecific because we get we're bringing two cells together. As you would see on the left panel, current bispecifics are in again a single action. They are bringing two cells together. One is an effector cell to eliminate cancer. In that regard, we are not bringing two cells together for at all. In fact, our molecule is, we would say that we are practicing social distancing at a cellular level. We have right now in the fundraising, so we are currently seeking both partners as well as investments. We are actively we have a collaboration with Ajinomoto, and we're looking for partners who work with us for new non-conjugation technology, new liquor, new payload, as well as new format. And we are currently raising funds and happy to talk to you often regarding our current raise and how the run looks like. I'm happy to take any questions.
Thank you, Shiva. That was terrific. I think we're going to, given that we're running over time, we're going to continue to our next presentation, which is Triumvira Immunologics and Joshua Carl.
Hi, good morning to our friends in Korea, and thank you to the conference organizers for the opportunity. Similar to Shiva, it looks like I'm having a bit of an issue sharing my screen. Great.
We're seeing now. You're starting to share your screen.
Excellent. So as you know, despite the transformative efficacy of existing cell therapies, a significant unmet need remains, primarily in two areas. One is toxicity, usually fatal cytokine release syndrome and all the debilitating neurotox. And secondly, there's limited settings where existing cell therapies work. It's just heme malignancies, really just B-cell malignancies within heme malignancies, and not solid tumors. And Triumvira is developing the T-cell antigen coupler, or TAC, as we call it. That scientific rationale and early data may be able to overcome those limitations. Our technology, it's not a CAR, it's not a BiTE, it's not a bispecific or other T-cell engager. We're pursuing both autologous and allogeneic approaches. And across multiple solid tumor and heme malignancy models, we have demonstrated TAC T cells outperform CAR T cells in models of interest to many parties. This has been externally validated by potential pharma and biotech partners. We have 3 ongoing evaluation agreements with leaders in the field that have been the source of this external validation, and it's great to see them observe in their labs the same data and activity that Vera is observing in our labs. As I mentioned, we're developing autologous approaches, and we're aware of some of the changes with autologous manufacturing. So we have entered into a collaboration with for their automated, potentially point-of-care manufacturing system. We believe, given our rationale for active solid tumors and the high incidence of solid tumors, that this will enable us to deliver our TAC T cells to solid tumor patients. All of this is built upon a very experienced management team and a CA, which we closed about the middle of last year. It was co-led by venture arm led by Bayer and also North Pond Ventures. So we put together what we feel is a very strong syndicate of both strategic and traditional financial investors, as well as strong representation. We have investors from the US, a couple from Asia, as well as insiders from Canada. We're, as many of you know, the best time to raise money is when you have some cash in the bank. So we're currently taking a crossover round, which we hope to close in the middle of this year, 21. And given the exuberance for life science investment currently, we want to be in a position to go public as early as next year. Our TAC platform, it really is a true platform that we believe in the broad applicability of it. And we're trying to set up a number of potential milestones and value inflection points over the next 2 to 3 years with this pipeline. Our lead program is a TAC targeting HER2 overexpressing tumors. We plan to enroll breast cancer, gastric, ovarian, and potentially sarcoma. We're filing the IND in the next couple of weeks and hope to have our first patient in potentially of April of this year. Additionally, we have world-renowned cancer centers being a part of this clinical trial. As you know, HER2 is a validated target, which we believe will be a great clinical proof of concept for our TAC construct. After demonstrating proof of concept, our plan is to invest aggressively in our allogeneic approach, filing our first allogeneic IND at the beginning of next year, and then also pursue aggressively novel solid tumor targets. So let me tell you a little bit about our TAC construct. It's comprised of 3 binding domains. The first is an antigen binding domain, which could be any antigen of choice, CD19, BCMA, or any novel antigen. And this binds to the tumor cell. Really the special, if you will, are the second and third binding domains. The second is a CD3 epsilon binding domain, which pulls in the full T cell to the site of the tumor. And the third, a transmembrane domain, which has components of the CD4 co-receptor and also binds to important kinases like LCK that an immune response, as well as shutting down an immune response. So the whole gas and brake aspect of a natural response. And what Vera and our TAC technology seek to achieve is to co-opt nature's immune response, which has evolved over millions of years, to effective response at eliminating foreign pathogens and tumor cells at the same time, not harming the host. And so here in the middle of the middle graphic, we have nature's T cell receptor with all of its complexities, a number of peptides, ITAMs, either CD4 or CD8 co-receptor to LCK, which on the left you see that Vera is seek to mimic this from a biological perspective and actually only activate through the T cell receptor, which we think is a very differentiated mechanism of action, very different from the CARs, which are, while again really seek to replace a natural immune response with activation domains, co-stimulatory domains, which we believe to some of the unfavorable toxicity that I mentioned earlier, which are likely caused by tonic signaling, which essentially means there are always signaling at some baseline. They're on or very on. They're never really off. Triumvir's TAC construct doesn't have any activation or co-stimulatory domains, and because of differentiation, we've observed preclinically to characteristics that are improvement, especially tumors versus CARs and existing therapies. So no premature exhaustion, more of a memory phenotype, and improved T-cell persistence. As I mentioned earlier, we've demonstrated this across several heme and solid tumor models. This table is a summary. I'll just highlight one, our dose response study, which at a very low dose of 3 million T cells, we see complete tumor regression, and half of that at 1.5 million cells. An example, cadence of data slides to highlight, further articulate, Triumvir's differentiate. So here with our HER2 program, we have ovarian cancer solid tumor model. On the left, you have efficacy, and the graph on the right is safety. The green line on the left is the control, so the tumors grow uncontrollably. The red line in the middle is a second generation CD8 CAR. Second generation is currently the state of the art in the field. We have this data with 41BB called it didn't perform as well. So you see some stabilization of disease in the red line. But then if you look at the safety, at significant toxicity, the animals are losing up to 25% of the weight, which, as you know, is which you need to use. But our CSO wanted to get some long-term data and put them in a mouse ICU, if you will, to nurture them. Back to the SD graph, in blue, you see the TAC cells have immediate and complete regression of the tumor, but at limited toxicity. Question becomes, why is that? And here we have biopsies of tumors on the left and healthy tissue on the right. This is lung. We also have cardiovascular. In the middle in red are CAR-Ts. On the top in blue are TAC-Ts. If you look at the CAR-Ts, what we believe is occurring is they're coming into the lung, it's the first test organ, firing, and they're misfiring because there's no antigen present. So that, eventually the CAR-Ts will reach the tumor cells, but there's fewer tumor cells, the ones that do work, and that's why you don't see the intended effect. But top at the TAC-T cell, they come into the lung, present in the lung, but they're not firing. They don't fire until they reach the intended target tumor cell. They proliferate, they activate, and this is how you see that strong regression with little to no toxicity. Another biomarker slide from that same study. Here we have a number of cytokines. Notice log scale. CARs again, TACs in blue. And we see occurring are CARs are just producing and hammering out cytokines 100 to 1,000-fold higher than the TAC T cells. And there's a train of thought in the field that patients must endure these high cytokine production and toxicities to have any efficacy. Well, we really beg to differ because in this model and in other models, we see the TAC T cells are having that activity, not the CAR T cells. As I mentioned, we're starting shortly our phase 1/2 TAC HER2 program. We plan to stratify, it's a basket study that we plan to enroll multiple tumor types, as I mentioned, breast, ovarian sarcoma, but also stratify by level of HER2 expression. So 3+, 2+. After our dose expansion phase, we intend, if the data looks good, to look at 1+ and HER2 low expressors. Because as you know, similar to an ADC, HER2 for us is a docking station. So there is some rationale that we could have activity in HER2 low expressors potentially. Also, as I mentioned earlier, we have an allogeneic approach. We're pursuing gamma delta cells as a healthy donor source for our allogeneic TAC-T. We believe to date we've demonstrated preclinical proof of concept with our gamma delta TAC-T cells, meaning our gamma delta TAC-T cells are performing in vitro and in vivo the same as our alpha beta TAC-T cells were at a similar time point. Here we're just showing in vitro data, but we are sharing the in vivo data under confidentiality, but we're very encouraged by this and intend to file the IND early next year. This is a high level and a quick introduction to Triumvira and our TAC technology. I appreciate the opportunity and happy to take questions.
Thank you, Josh. That was an equally impressive presentation. I've been very impressed by the quality of what we've heard tonight, or tonight here in the U.S. or in the morning there in Korea. But it does sound like there's some real breakthrough status type drugs and innovation that's going on with this group of presenters, and it should be encouraging for anybody looking to partner or to invest, frankly. So our last presenter, so thank you again. Our last presenter is Eric, not sure I can pronounce that last name properly, so I won't butcher it, but he's the president and CEO of PDC Line Pharma. And I think this presentation has been pre-recorded, so I will ask KoreaBIO to please start presenting it.
Tell you that I'm sorry of not being present live, but due to the time difference, I was not able to be present and I had to record my presentations. So let me present you PDC Line in a nutshell. So we are a cancer vaccine company based in France and Belgium, and we are a spin-off from the French Blood Bank based on the discovery of Joël Plumin, the scientific founder, when he was in academic lab associated with the French Blood Bank, where he has identified and characterized a very rare leukemia affecting the plasmacytoid dendritic cells, which is the small fractions of the dendritic circulating in the blood that have very interesting property. He was the first one to characterize this leukemia, and based on the blood, on the PBMC of these patients, he has developed a GMP cell line and a clinical and a master cell bank approved for clinical use by the French authorities that we are still using today as a vaccinal platform. So he has been founded in 2014, and we are now a team of 25, 26 people. In terms of funding, we get close to 32 million euros in financing, 20 million in equity, and the rest in non-dilutive money. Our technology presents three key benefits or advantages. First of it, it's a cell line of dendritic cells, and the cells has been immortalized due to the leukemia, and it's extremely easy to expand. I will show you the manufacturing process. Second, it's a plasmacytoid biological property, and it's a very highly potent antigen-presenting cells, able to expand the antigen-specific CD8 T cells. And it is a versatile platform. We just have to load the cell line with GMP peptide representative of tumor antigens, and we can address potentially any kind of solid or liquid tumors. So the company is a clinical stage. Phase 1B in melanoma has been completed, and we are now on go in the phase 1/2 in non-small cell lung cancer. So the pipeline is now in France, Belgium, Germany, and we are opening Netherlands. And the neoantigens is in preclinical stage. Here we are looking to identify and characterize specific tumor and neoantigens induced by the tumorogeny of the specific tumor genetic modifications of the tumor. Important element, we have signed a first licensing deal in early 2019 with LG Chem that provide us a good contact with the South Korean ecosystem. The deal were concerning the PDC lung. We licensed the rights, we provided the right for development and commercialization in Asia. We granted exclusive license in South Korea and options, exclusive options in other Asian countries. The total deal value is $123 million plus significant royalties on net sales. Another key asset of the company is definitely the management team. We have in the team people with very strong experience in cancer vaccine. Our medical director, Shanna De Bruyne, come from GSK vaccine, and she led the registration clinical trials of the cancer vaccine in melanoma and lung cancer of GSK. Claude Dédry has a very large experience in cell therapy and CMC. Is the head joined full-time the company, and myself, I'm, as we say, a serial entrepreneur. I've co-founded several biotech. I've been involved in several exits. I sold my first company, Music to Enzyme. I've been involved in IPO and raised more than 130 million euros in my career. In the governance body, in the board of directors, we have as well very senior and experienced people. The chairman of the board, Monder Majoubi, is the former executive VP of AstraZeneca Oncology, is an oncologist, and of Genentech. He's currently the CEO of Innate Pharma, company, a public company in France. Jean-Paul Priels, who is an independent board member, is the former global R&D director of GSK Vaccine. He's as well an investor in the company. And we have, of course, representative of the investor, including Song-Woo Lee of Korea Investment Partners, who is the new Korean fund that joined the last round of financing that we have completed beginning of last year. In terms of achievement, the company has been quite busy over the last 18 months. I will present you the key achievement made. In January 2019, we get the GMP accreditations for the QC and the release of the vaccine from the Belgian authorities. In March 2019, we signed the licensing deal with LG Chem. In June, we get the authorization to launch the phase 1/2 in non-small cell lung cancer in France and Belgium. In August, we get the arrival of Dr. Shanna De Bruyne as a new medical director. In December 2019, we closed a 20 million B round of financing led by Korea Investment Partners. Two other South Korean investors joined as well, the syndicate of investors. In February, we announced the administration of the first patients with the cancer vaccine candidate. We had the arrival of the president of the board, Monder Majoubi, in June. And finally, we announced in September the dosing of the first patient with the high dose in the cancer vaccine PDC lung. And that's showing the key element and the movement that the company has been involved in over the last 18 months. In terms of technology, let me describe you the manufacturing process. As I said earlier, we have a master cell bank where the cell line is stored in bag in liquid nitrogens. And we have developed from this master cell bank several working cell bank. And we just have to take a bag of the working cell bank and seed a single-use bioreactor. The cells can grow in this bioreactor. We have 1 liter and 10 liters bioreactor. The cells grow in suspensions in a synthetic medium, serum-free. And after 9 to 10 days of culture, we have billion and billion of cells. At the harvest, we just have to split the cells in bag according to the number of peptide we want to load on the cell line, on the HLA present in the membrane of the cell line. And then after the loading individually, we pull the different bag and the cells. Then we irradiate the cells to stop the proliferation. I remind you, it's a leukemia cell line that we are working on. Then the cell irradiated and loaded are put in a specific container, the technology of aseptic technology, and stored in liquid nitrogens. And when the patient is ready, we just have to tow the cell and with a syringe to take the suspensions of the cell line and infuse to the patients. Then the cells will do the priming of naive T cells, antigen specific, and expand memory T cells that become cytotoxic CD8 T cells that will migrate to the tumor microenvironment to make the job, which is to kill the tumor. So this manufacturing process is extremely scalable and low cost. Another key benefit of our technology is the potency. You see here an illustration of the results of an in vitro study we have conducted with the cell line loaded with a specific antigens, which is melanin. And we have measured by FACS Dextramer the expansions of CD8 T cells specific of the melanin after 21 days of culture with our cell line loaded with the melanin, and compare with autologous conventional dendritic cells or allogenic dendritic cells. And as you can see here, we are definitely more potent. We are 20 to 200-fold more potent than the classical approach. So the cell line is very potent and low cost to produce. We did study on neoantigens. As you see here, an example of experiment we did as well in vitro, where we have selected 2 neoepitopes where the mutations in red has been characterized. And this epitope has been identified in the blood of a melanoma and lung cancer patient. And this peptide has been published in the scientific literature. And we loaded the cell line with these two epitopes and put in contact with the blood of healthy donors. And as you can see here with the CD8 of these healthy donors, after 20 in the right, 14 to 21 days of culture, you see in red and purple these nice expansions of the clone of CD8 T cells specific of each of these peptides. We have close to 150-fold increase for both peptides, so showing the potency of the cell line to expand whatever shared antigens or neoantigens. Knowing that in the blood of it's 1 of 1 million CD8 in culture. In terms of IP, we have 3 key pillars in terms of IP strategy. First of it, we have the proprietary cell line, which is unique worldwide. We have this master cell bank, which is clinical grade, and we are the only one with the French blood bot that can use it. We have patent, of course, 2 family of patents that has been granted, protecting the cell line and its applications, and a third one that has been filed more recently, which is in PCT process on the genetic optimizations of the PTC line. And we have all the know-how and data related to the manufacturing, the immunomonitoring assay, and the preclinical data. Okay, let's pass now to the result of the melanoma feasibility study clinical trials. So we did study on 9 patients with advanced melanoma, phase 4, after 3rd line or 4th line of treatment. The cell line was loaded with 4 different antigens, tyrosinase, GP100, melanin, and MAGE3. The result was definitely promising. The primary endpoint, which was the safety, was acceptable. The we didn't observe any allogenic rejection detected in any of the 9 patients, even after 3 infusions of the cell line loaded with this 4-peptide. And we have observed as well significant antigen-specific CD8 T cell response directly detectable in the blood of the patients, in some of the patients related to some of the antigens, without any in vitro expansion. So showing that the vaccine can work in this setting. And that despite we just infused 3 times, only 3 injections, and we didn't co-administrate any checkpoint inhibitor. And we know that synergy with checkpoint inhibitor and our vaccine, and as melanoma patients, melanoma is a very hot tumor. Contact with the PBMC of these melanoma patients, and after 1 week of culture, you see that we can expand by 15-fold the CD8 T cell specific of the antigens, which is melanA. When you put the checkpoint inhibitor,
Unfortunately, we seem to have lost the video from our last presenter, PDC Line Pharma. But I do want to let everybody know that we are going to be making recordings of all of these presentations available, and they will, of course, be much better quality. They'll be posted to KoreaBIO's YouTube BioTV page, and so you should check back there for their availability. But I just wanted to wrap things up briefly and say thank you to all the presenters and to the organizers of the event, KoreaBIO. This was a terrific set of panelists and presenting companies, as I mentioned, and a terrific panel discussion that preceded it. I hope that everybody stays safe and healthy, and then we can all convene together in person next year at JP Morgan and see everyone soon, either here in the US or in Korea. So with that, I will say thank you and good evening.